Hello, everyone. My name is Josh Ludwig, the U.S. Commercial Director at ScaleReady, and thanks for joining us for an extremely exciting panel featuring an all-star cast of industry leaders who have come to help us discuss the key decision points and considerations for intelligently scaling up cell therapy manufacturing. The industry has established that cell therapies are a viable treatment option, and now we must refine and standardize our manufacturing processes so that they will meet patient demand, not just in the immediate future, but for a future where cell therapies become primary and secondary treatment strategies. To begin the session, I'd like to play a short video to better introduce you, the audience, to ScaleReady and the scaling challenges that our platform is addressing.
When it's time to scale up from cell and gene therapy research to a clinical treatment that can help thousands of patients, will you be ready? Can you reliably predict how your cell expansion process will perform when generating 10 million, 100 million, or 100 billion cells? Will a complex bioreactor with a big footprint give you the capacity you need to treat more people? We're ScaleReady, a partnership between 3 leaders in cell and gene therapy. We're thinking ahead with a bold new vision of cell therapy manufacturing that scales intelligently. Our platform rethinks 100 years of cell culture assumptions to create a radically intuitive solution centered around the G-Rex closed system bioreactor. Our ingeniously simple bioreactor gives cells unlimited access to nutrients and oxygen, creating a unique zone of diffusion where they can communicate easily and multiply naturally, all without the mechanical motion and constant feeding of complex bioreactors. Simplicity solves scaling up. The same design scales reproducibly from 10 million cells to 100 billion in escalating G-Rex sizes. It also solves scaling out. With 1 G-Rex per patient, G-Rex bioreactors are a more practical, cost-effective, and predictable solution for serving large patient populations. Treating larger patient groups is as easy as adding more devices. You can fit dozens of G-Rex bioreactors in the same space as just one complex manufacturing system. Over 500 organizations and 150 clinical trials worldwide are using ScaleReady solutions, demonstrating how our thoughtfully designed workflow is the most powerful and versatile platform for cell therapy manufacturing. Wilson Wolf's G-Rex closed system bioreactors are the advanced engine for the whole workflow. Biotechne provides quality-driven GMP media, activation reagents, cytokines, and non-viral gene modification technologies. Fresenius Kabi provides Lovo and Q cell processing systems that efficiently bring in cells from patients and process out cell therapies to help them. We've thought ahead to where the technology needs to be for cost-effective, practical scale-up and scale-out. So think ahead to where you need to be, and you'll discover that ScaleReady is already there.
All right, hopefully that video played. I'm not sure if there was any technical difficulties. Can we get confirmation on that from the support team?
I couldn't hear it.
Okay. Yeah. Well, so we'll just move forward. At this point, I want to introduce you all to our panel for today. Go around the room. We have James Bowling, the cell therapy process development lead at Precision Biosciences. Kelly Ganji, President and CEO at Amplify Bio. Tim Moore, President and Chief Operating Officer at Pact Pharma. Mike Paglia, Chief Operating Officer at Elevate Bio. And Amos Tsai, Head of Late Phase Process and Product Development at Autolus Therapeutics. And our session chair today is Juan Vera, who is the Chief Scientific Officer at Marker Therapeutics. And so now I'd like to hand things off to Juan and remind the audience that we do have a Q&A chat function, so please participate throughout the event. Thank you. Juan?
Thank you, Josh. I know that there were some technical difficulties with the video. Perhaps if we have time, Josh, at the end of the session, we'll hand back to you for you to play the video. So hopefully by then we will have this technical issue sorted out.
Sounds good.
Okay, well, thank you very much for the introduction, and it's really a pleasure being surrounded with such a panel of experts. I think that it's definitely a very interesting time for cellular therapies, and it's a field that continues to evolve and it continues to change. I would like for today's conversation to be very dynamic. Also, questions that will be coming from the audience. So feel free to participate as you feel fit. Why don't we kick off the conversation with perhaps an open question related to your sort of personal experience in terms of translating a process from early stage into the clinic. I think that we all have had different challenges in that regard, and perhaps different lessons learned. So maybe I hand over and open the question with Mike. I know you have a specific experience and related to translating process that have been early into the clinic. And what would you like to share in that regard, Mike? What would be your lessons to share with the audience in terms of things learned from translating early stage into the manufacturing process?
Thanks, Juan. So I think at the highest level, the excitement in the cell and gene therapy field is at the rate at which these products can get approved. So getting it right the first time is very important, and making those scale and those translational decisions very early is important as well. So something we've done here at Elevate Bio is build that technology capability to enable cell and gene therapy companies to be able to accelerate that, but also to translate those therapies very quickly from process development, or from concept, I should say, into process development and into GMP manufacturing through that technology. So thinking about the scalability of the process and the translation, justifying those scale down models is very important. So making sure that you select a process or have a capability to be able to pivot very quickly from that early stage clinical concept, clinical trial into process characterization, and frankly, get ready for PPQ very early on is very important. So many of the devices, bioreactors, systems may not necessarily have that capability, but certainly one that does is the G-Rex device, and thinking about having that ability to not only get into clinic very quickly with a scalable device, but then use the technology, justify those smaller scale down models, even in a high throughput way, which looks like the future for what G-Rex is working on and Wilson Wolf, is very important. So what we see is that translational time, that process understanding through characterization, and making sure you have a device and a technology that you can have that ability. And we see it, I've seen it many times where CMC is often behind because these products get proof of concept and they're very transformative very, very quickly. So making sure you have that ability in whatever selection system that you choose is very important.
Thank you, Mike. And your experience is quite unique. I mean, you were earlier on working with Bluebird, and you have really a lot of experience advancing some of these products, right? And now as part of Elevate, I think that perhaps if you can tell the audience and the panelists, what is Elevate? Because maybe some of us is still not fully familiarized with what Elevate is and how you can do the contrast in your previous experience with Bluebird and now what you're doing.
Sure. Yeah. As I said, Elevate Bio is built on the fundamentals of a technology company where we can power cell and gene therapy therapies with our technology. So thinking about the ability to have an infrastructure here in Waltham that has process development capabilities and GMP, our team here at Elevate Bio can take the technology that we've developed in-house and work with, whether it's our own companies from a portfolio perspective or other clients. So we have a manufacturing business and process development, capability here where we like to work with partners in the cell and gene therapy industry to come in, work with us. We can very quickly translate those products through process development and manufacturing, but also get access to other technologies that we're developing in-house, such as we have gene editing technology and therapeutics with our company called Life Edit. We also have inducible pluripotent stem cell technology. So thinking about the world of autologous to allogeneic, but also the manufacturing processes as well, and using G-Rex devices and other devices and technologies to make cell therapy products as well as viral vectors. And again, translate them very quickly from development all the way through GMP manufacturing. So really, we have a lot of technology at the company that we're trying to embed and help accelerate, but also democratize that, making sure that if someone would like to use that, if they build their own facility someday, to be able to help enable those companies in that industry to be able to advance the field, not necessarily always keeping it. We like to expand and grow with the technologies in the companies, and that's our intent, but also to be able to make sure that we can enable the field to advance through our technology as well.
That's great. Thank you, Mike. That's very interesting. Kelly, I know you come from a very different background as a CMO, and you have a lot of experience in this regard, especially when translating process from early stage into as a CMO. What is your take on the challenges into taking a new process on board?
Yeah. Well, thanks for that. I'd like to underline what Mike said. I mean, I think we've seen the same level of fluidity or continuity in a number of clients that we worked with. And I think, if we go back in my 11 years at Cognate, I think we might have been some of the first groups using, G-Rexes as they were hitting the market. And so I feel like, we grew up a little bit with Wilson Wolf in terms of their offering as well. And it's been really nice to see them scale up and scale out the offering, on both sides of that. And if I were to put myself in the developer's shoes, as we did many times when we were in the CDMO world, the last thing a developer needs to be worried about is the actual platform. They've got so many other things that they're worried about, whether it, range from cell counting to, managing, aspects of fill finish or other things. To have a platform that can go from some of the earlier stages up, in clinical development all the way through commercial was really, that was one of the easier parts of the development process. There were, given that there are so many other parts of the puzzle that are not just little things to deal with, but major things to overcome. So, and as we are all accustomed to, the process is the product. And so when you've picked a platform like what your cells are actually growing on, you don't want to have to go to the agency and defend why you're getting less viability from cells, and when you actually look at it, maybe they don't bind as well to the carrier that you've selected. So having that continuity across early and later stages was and is and has been a really great part of working with Wilson Wolf over the years.
Great, thank you, Kelly. Maybe if I can now jump the same question to James, which I think that you probably have a different take in terms of what it takes to translate a process into an early stage into the clinic and the manufacturing piece. So what will be your message or lesson learned on that regard, James?
Yeah, thanks, Juan. I think there's a number of aspects that are important to put out there. But I think first and foremost, to echo what Mike said, getting it right the first time is critical. And so, for us as a allogeneic CAR T company, we were looking for something that was going to be able to take, research scale, open processes and scale it up to, tens of billions of cells, without all the headaches that come with many of the platforms that are capable of doing that. And so for us, looking at Wilson Wolf compared to other types of bioreactor systems, there were some clear advantages from the start. Having the ability to scale down, as well as scale all the way up to the 10s, 20, 30 billion cells without issue, was critical from the start. And then looking at how do you implement some complex gene editing steps into an overall process, and how does the the activation of the T cells as well as the expansion of the T cells, how does that fit in, and how do your unit operations really match up when you're thinking about the devices that you're using? And so when we were looking at many of our CQAs and process performance metrics, there was just really not a big not really a way to make some of the more complex systems work while maintaining product quality in a suitable way. So I think for us, we had to make a decision early on to go with one platform over another, and making sure that we made the right decision was critical. And we ran countless experiments and head-to-head studies looking at what made the most sense. But ultimately, the simplicity of scale, scaling the process, as well as the fitting in complex unit operations, made it a pretty clear decision.
Right, great. Thank you, James. And you did mention something very valuable in terms of like when making that decision of a platform. And that's something that I will come back in a moment and perhaps get a take on that from the different panelists. Perhaps, Amos, if you can give us what is your impression. I know that you come from a field that is similar to James. You're working in Autolus, which is this off-the-shelf platform. And so what is your take on this challenge of taking a process into CMC and into the clinic? Amos, you might be on mute. We cannot hear you.
Sorry, my apology.
There you go.
Okay. So first of all, I just want to thank you for the opportunity to be participate in this panel. So just going back to the question about the challenges, I see that. Okay, so just going back to the title of this discussion here is really about how small decisions have major implication for intelligent scale up and out. Clearly, that's really the challenges, right? We all know that these small decisions that have been made cumulatively during early development inevitably would become impedance to our ability to scale up or scale out. So then really understanding how these various small decisions have to do with the fundamental science in terms of activation, proliferation, and eventually product performance. And then how do we actually would streamline them out, right, by using a particular format versus another, Wilson Wolf versus Miltini versus whatever else, right? Some sort of a technology would lend themselves more for us to streamline these various small decisions that have been made cumulatively, with an eye toward their potential impact of lack thereof on process performance and product quality. That's one challenge. A second challenge really is that we know that the technology that we're working on right now are not going to be the technology we're going to be working on 2 years from now. In the same time, as a result of our need to scale up and scale out, we need to aggregate these technology into a whole, right? Into a system. We have different components that we're working on that we know are going to evolve over the next two to 5 years. And then we're building a system around it. And how do we actually link them together so that they're positioned to adapt to changes over time? Even within the Wilson Wolf world, we know they're working on innovation that would help us do better. So that's another level of challenge that we need to be aware of. So that's kind of what I'm seeing right now is that, how to actually clean up the various small decisions that have been made in a way that is sensible. Pick a platform that will allow us to clean up these small decisions. And then while we're trying to aggregate the components together into a system, make sure that it is flexible enough that when these individual component is improving, that we can still leverage that improvement for life cycle management.
Thank you. You speak a lot of things that I find very, very interesting. You talk about this evolution of the process, right? And quality and life cycle, right? And I would like to dig in more detail in some of those components, which I think the analytical and the understanding of the scientific mechanism of action of the product is a critical piece, right? So I will come back to that, Amos, if that's okay, later on, so that maybe we can go into more detail. Kelly, you had a comment.
Yeah, I wanted to and I feel a little bit maybe a little conflicted with the statement, but I wanted to make sure that the audience members realize, and if you've been in the field long enough, what we were faced with, at least from a CDMO perspective, is we had a lot of clients coming to us with technologies that were still being grown in flasks, and it was like, we want you to maintain sterility because this is a one-off product and we've got a patient that's ready to get dosed and they have no other options. Don't contaminate this, but you're going to have to take the lid off and use a manual pipette. So, I mean, like Wilson Wolf really changed the game in that regard for those of us who were trying to maintain and manage, at the time, tens and maybe, if we were lucky, hundreds of products, in an environment that didn't give people like myself heart failure every time we were going in to change media or add supplements. Like, this was a real game changer, and I wanted to make sure that was stated, and it was not just that they gave us a new platform to operate on, but they continued to grow. I mean, like, honestly, when John was talking about a bioreactor, and I was thinking, okay, well, we'll have that in many years from now, and we see that in the market now. So it's really impressive how they've managed to continue with this technology and continue to develop it. Meanwhile, still meeting the demands of the industry. When it was, I want to say it was 5 to 7 years, somewhere between 5 to 7 years ago. The demand went from 10 to 10,000 almost overnight for G-Rexes, and Wilson Wolf pulled it off in a pretty remarkable time. So, I know it sounds like a major plug. I'm actually not in the CDMO business at the moment, so I'm not plugging him. But I just wanted to give kudos where kudos are due. They really did. They really changed the game for us a while ago.
You're right. Thank you, Kelly. No, I think that is really valuable. And I think that it definitely, for many of us, we have had first-hand experience with this platform. And I think that it's something that definitely the simplicity of it had allowed the acceptance by the field, right? I mean, I think that reproducibility and scalability for sure is something that it is very critical when translating a new manufacturing process, right? But I think that also goes hand in hand with the thing that Amos was mentioning a moment ago, which is that understanding of the product, right? The scientific premise, how the product actually works, because the process will evolve over time, right? I mean, I think that you have all seen it, right? And being able to have that common thread, which you can call analyticals or potency, to show continuity as the process evolve, is something that is very valuable, right? Especially when these are related to the FDA, right? In terms of how the new processes are being advanced, right? So, I agree with you, Kelly. I mean, I think kudos to the people from Wilson Wolf, because I have seen a lot of that back and forward with the scientific community, where a lot of the design actually had been fed back to the devices, right? So it's a really bidirectional relationship, which is unlike any other group. And I think that that's part of the reason the devices work so well, because they have taken at heart a lot of the feedback that they have here from many of you in the panels, right? Say, this is the thing that experts or people would like to have, and they're ultimately incorporated, which I think is really what makes it different, right? Okay, but so I guess, Tim, you missed the first section of the introduction. Perhaps if you can very briefly in a minute or so say something about yourself, the work that you currently are doing, and then I pivot into that first question in terms of what is the challenge in your experience you have seen translating a process from the beginning into a CMC. Yeah, so what is your take on that? Yeah.
Sure. So just a brief introduction. I've been in the biotech industry for over 35 years. Been in cell therapy for over 5 years. Haven't spent a lot of the time initially at Kite, growing and expanding out their operations in the US, Europe, and even in China, where we did a joint venture. Prior to that, I spent many years at Genentech on the head of the global operations there. So I really, even though I love Genentech, Roche as a company and working there, I got super excited about the cell therapy industry. And that's why I jumped over to Kite to help grow and expand that company to make a difference. And now that was, it's primarily focused at Kite is autologous products, but very specific. You make one CD, for example, CD19 target to gene edit each individual patient's T cells for whether it's leukemia, they have a leukemia product, a lympho product. Here at PACT, where I'm heading this up to scale out as well, we are focused on the next level of personalized medicine and autologous, because we're, for every patient's own solid tumor cancer, we're identifying specific neoantigen targets to gene edit into the patient. And we identify those by, we have a very proprietary, what we call our discovery and verification process of taking the biopsy samples as well as PBMC samples from the patient to identify which targets look the best. But we don't just predict, we also do a verification by showing they can attack the tumor and do something to it. And that to me is super unique and innovative here. But one thing that, being in this industry, not as long as I predict some of you, but I still feel the cell and gene therapy is in still early stage, and so the technology development is still going to grow. And what we need to be able to do is be able to continuously improve and be flexible. And I think Amos, you had brought up the analytics, and I totally agree with you. One of the key things in our space is analytics. And when I say that, it's end to end. Let's just say, I'll take it for like a CAR T. You need to understand each patient's incoming apheresis versus their clinical, and then try to identify product characterization across the line to see how, because different, even though you're treating, let's say, the same cancer, different patients' cells are going to be different and they may act different. And to me, as we scale up this whole industry, the analytics is going to be a very important part. And when I think about it from a production standpoint, one of the challenges I see is how can we get better at doing the analytics real time, not just, because you can't always pull as much samples as you'd like, because then you're taking out cells and you may not get the growth level that you want. So how can you do it in process, whether it's just cell count, or can you do flow cytometry? You know, I could go down and spend a lot of time. That is a technology, I think, that is a challenge today that I would love to work and see that come along. And as at Kite, we didn't use the G-Rex. Here at PACT, we are using the G-Rex. You know, whether it's bioreactors, or I'll call it that, do the, as that video showed, doing the shaking up and down, or the G-Rex, which is just stable. Both work. Now, which ones work the best for the particular product, going back to analytics, is the one that is really important that we be able to identify. And also, as we advance and looking at everything out there, how do we bring this fully automation into connection with each other, both from a process standpoint, but also a data management standpoint? To really advance it is going to be key to growing out the industry. Because as I've scaled out to treat thousands and thousands of patients at my time at Kite, it also took hiring lots of people. And that's not a bad thing. However, that can affect scaling. So if we can really get this to be more fully automated, I think scale out will become easier and easier over time.
Yeah, no, that's super interesting. Actually, I can plug one of we did present a poster in automation using the G-Rex, which again, you've seen it. Yeah, it's very early work, but I think that it's something that we're working on in collaboration with ABB Robotics, right? And I think that it's something that it could be an alternative, right? I mean, I think that we all have learned by experience that sometimes these small things of the force and how the culture is being handled can actually influence the actual outcome. But one of the things that for me, at least, is very rewarding is that we have gone above the point of getting enough cells. I think for the most part, most groups are now able to manufacture sufficient cells, but now have gone from the point of quantity to perhaps quality, right? And like, are we making the cells of the right quality of the attributes that we actually want to have? And are they correlated with the attributes that will perform the best in the clinic? And I think that the field is still learning what aspects are important there, right? But it's still, there is a big gap, right? There's still things that we're learning in terms of what are the desired attributes in that final product, right? But Kelly, you wanted to mention something. I see you.
Yeah, I wanted to sort of underline again something Tim said, and to point out that the reproducibility is really the focus of the FDA, right? So they want to see that if you're making 5, 50, or 500, that they're the same, and that you can demonstrate that. And to that point, I won't mention client names or even allude to the fact that they were a client of Cognate's, but I do know of companies that have gone through early stage, gotten to a point where they said, okay, we're going to scale up. They go to a larger container and the product is different. And so then they come back and have to scale down. And when they go back to the FDA and say, okay, here's our commercialization plan, the FDA says, that's ridiculous. No, you need to show us that you're scaling your process, not just that you can, grow 500, T50s or T25s. You need to actually show a scalable process, because you can't establish comparability across 50 T50s. I mean, this was years ago, but it just it's something that we've seen and have been very pleasantly surprised with over the years, of how having Wilson Wolf be able to scale up with our clients in different containers was something that. And as I said before, there's so many things that you have to worry about when you're developing a drug. The last thing you want to worry about is the container that you're actually putting them in. So it allows you to focus on the important things. And I've mentioned previously about fill finish, but you can make a wonderful product and it can grow great. But if you have a process with fill finish, half of your product can go in the trash can. So when we talk about things like that, those are the important things that we should be focusing on, and not having to worry about from going from a smaller container to a large container as you scale. And my feeling is that we've been able to do that pretty well with the G-Rex.
Yeah. Absolutely. I mean, I think that one of the advantages of the platform, without a doubt, is how it can be easily scaled up, allowing really to be able to create models much, much faster, right? But let me go back to a couple of things that were mentioned in the initial question, and I will go back to James. Some of the things that you mentioned earlier on, you said something about making the decision on the platform, right? And that I would like to challenge and maybe ask a little bit deeper questions. When do you make the decision, and how do you make the decision on the platform? I think that those are very important things. So perhaps, James, maybe we start with you, and then we go across the table. What you can tell us about that?
Yeah, I mean, I think from our experience, it's difficult for any individual program. And so we're working with four separate INDs. But we've really focused on developing a platform process that can be kind of utilized regardless of the CAR T program and regardless of the gene edits that are made. And we've been very successful with kind of that platform approach thus far. And I think that that's really a testament to making the right decision the first time. What we have kind of experienced is that, as we're moving some of these programs through kind of early stage clinical trials, making changes, even small changes to the process, do require quite a bit of complex comparability work that really stresses the boundaries of kind of the regulatory guidance and our quality system and our process development. Because what some people have already brought up is that the analytics are a challenge, and flow cytometry really doesn't give you all the information that we really need to kind of make that connection between the mechanism of action and some of these markers that we can get by flow cytometry. So I think having better potency methods is always been a challenge and will continue to be. I think I just this week I've seen some really exciting data internally on potency methods that may be kind of turning the corner towards something that we can run consistently to demonstrate that potency. But it's a challenge, and I think that getting back to making process changes without having a good product quality attribute that gives you potency, comparability is kind of nonsense. Obviously the viability needs to be above a certain level. Obviously, the knock-in percentage needs to be above a certain level. That's all great, but what does that really mean in terms of comparability? I think that's really why making a decision at an early stage and making the right decision with kind of the full view of how things are going to change. I think Amos made a great point that we know that both our understanding of the process as well as the equipment that we hope to be using is not the same today as what it will be in 5 years, and we need to kind of be cognizant of where we think the biggest gains in the process are going to come from, and kind of hedge our bets a little bit in that respect. So I think that the culture platform was kind of an easy one to say, hey, this looks like it's future-proofed in a lot of ways, and doesn't come with a lot of baggage of complexity and variability and lack of scalability that kind of alternatives obviously have.
Great. No, those are really, really great points. And you talk about the baggage, right? So making the early decision early. You mentioned that one of the platforms was perhaps an easy one because it becomes clear this is a lot simpler than other alternatives, right? And outside of that you refer in terms of simplicity, is there any other aspect that you look earlier on to adapt a platform? What else would you say?
I mean, I think what we saw is that just from the straight numbers, right? I mean, we talk about what works and what doesn't work. Well, when you have a relatively complex gene editing step, as we do, I mean, it is a single step knockout and knock-in process, but it's not easy. It's not easy on the cells, and I think having a platform that accepts cells after that process and allows them to expand at super high viability and really kind of thrive was, that was really the kicker. Beyond everything else, what we saw is that alternative methods could not reproduce the quality attributes and process performance that the G-Rex could. So I think that's the big one. Beyond that, I think that the ability to the cost of goods aspects and the ability to kind of optimize the efficiency of a plant overall, and thinking about how you design your suites and how you isolate individual products and individual batches. I think that with the G-Rex system, there are a lot of opportunities to really get the most out of your plant and your manufacturing system that I think you can probably do. And maybe Tim can speak to this a little bit with how Kite has run it, but you can do that with shaking bioreactor systems, but it's probably, there's something to be said for kind of the closed and scale up and scale out aspects of the G-Rex system.
Got it. Thank you, James. That's really well said. Tim, I think that you have been nominated by James. You would like to comment a little bit more on that, and if you can highlight about making that decision. When would you consider making a decision on a platform? When is early enough? And what attributes do you look into making a decision for a platform?
Well, decision on a platform, you got to take into consideration, are you in your pivotal trial? Because FDA will expect you to be somewhat, like, I'll just use an arbitrary number, 90% locked down. That isn't changing for what you're going to do to file your BLA. So to me, your phase 1A, 1B beginning of a pivotal study is the critical time. And that's why continuous improvement, really understanding your end-to-end analytical data, continuous improvement to modify and lock down your process by then is going to be really important from my experience. That said, the good news, my time at Kite, when we were advancing automation, even after we went to commercial, FDA was very open that said, if we can just show comparability, and I know comparability varies in different people's minds, if we could show comparability, they would view advancing the automation and, let's just say, the unit operations as a CBE 30. And I got very excited about that because I thought it was going to end up being a pre-approval. And we were able to advance some of that. So really early in your clinical study is when I think it's important that you really hone in on what platform are you going to clearly move forward with, so you can keep on your timelines. But like I said, and I heard James say, that's going to change over time. It's going to change over time.
Hey, Juan, maybe I'll just add to something that Tim was saying too, is for, the products sometimes we see coming out of early biotech or even academic settings, it's still surprising that they don't have the knowledge of what's out there, let alone the raw materials is a completely different story, selecting the right raw materials, but even with technologies out there. And I think what John had done, a long time ago is try to educate the field, right? It's not only through conference presence, but looking and working with the right partners to generate data. out of the G-Rex device and comparing it to other systems and showing the scalability, but in some cases difference, whether it's better or not, there were differences, and again, more healthy cells using the device, the way the diffusion happens and everything. So I think, that part of the industry, I've been on several panels recently where education is key to these innovators, whether make the right decisions for their process, but let them know that these novel therapies could get approved very quickly, and that translation to an approval or transformative therapy is critical to some of those early decisions. So having that educational aspect of what John and the team at Wilson Wolf have done over the years is also very important for the industry.
Yeah, absolutely. Thank you, Mike. Kelly, you're raising your hand. Yeah.
Yeah, just another 2 cents on that is that we to reiterate what Mike said. I mean, we did see a lot of people coming to us with an academic process, and no knocking on an academic process because a lot of patients' lives have been saved in that. But they brought the package to us and said, please don't change anything because it works. I just need to make 10 of these. And the other complicating factor to that is that many of the companies like this have made promises to whether it be the market or their investors that we're going to get this in trial and we're going to, get some confirmatory data. And then once we get that confirmatory data, you're going to write another check. So I would just, potentially suggest or ask or beg, whichever one you want, those that are out there starting these companies to plan for a little bit more on the PD side than you think. Just because a process has been made and put in humans doesn't mean that it's really ready for prime time. And so, if you think you're just going to flip a switch and go into humans, budget some more money so that you can actually maybe you can run them in parallel, like, Mike and Tim said, so you can develop that comparability so that once you actually do get that milestone, you can switch over to something that's a little more commercial.
Thank you, Kelly. You bring a very important point that I would like to ask later on. When and who should be in charge of doing that PD work, right? Because you talk about that studies coming from academia, and I tell you in academia, that's not where that work is developed, right? And I think that creates a barrier, right? So, and I can see by the reaction on the panelists, you all have faced that kind of gap. So I would like to dig a little bit more in terms of where that product development phase should be developed, because perhaps this is something that should be done even earlier on, and our colleagues in academia maybe should be looking into that with a little bit more attention, and I can speak that by experience. And I think we have gone across on this first set of questions. We do have a couple of questions that in the pool that we will put to the audience to get the reaction. Josh or Kelly, are we ready maybe to pull the first question that we have to the audience?
Yeah, it looks like we just sent the survey, Juan.
Okay, so let's just give some time for people in the audience to look through these questions. While we wait for that, well, I'm just going to give a moment for people to concentrate. Okay, well, maybe we can move on to the analyticals. I think that there was a lot of interest on that. Amos, you mentioned something about analyticals and the process evolution and how it actually changes. And Tim, you also echoed that, right? Like we need to understand that the process evolves. The question is, are we there yet? Do we understand what matters to be measuring at this point? I think that beyond cell viability and a couple of phenotypical markers, are we there yet in our understanding in terms of what matters? And what are your sense in terms of the evolution of analyticals? Some people talk about collecting samples, real-time assessment, and so forth. So maybe we start with you, Tim. What is your take on that? And we go around.
Yeah, to me, this is kind of tying to the PD. This is an important reason why to have, I always want my PD team to have an analytical development team part of it, because I'll just share some examples. Different patient cells coming in. Well, the CD4 to CD8 ratio may be different, or the phenotype makeup may be different, and not maybe, they will be different. And you see them perform differently in the manufacturing process. The analytics, really diving deep into the analytics, is important to understand if you can identify what are some potential manufacturing processes, or do you need to screen your patients differently, which I've learned over time, not being a MD, but I've learned over time that you get to where you learn things about patients that you got to screen the ones that are going to work best, at least in your platform today. And those are things that we work on here at PAC quite a bit, is really understanding how the patient's incoming cells, what that looks like in our manufacturing process, not only from cell growth and gene editing, but also what's the phenotype of the final product, and then also what do you see in the patient's treatment and outcomes, and those type of things. And kind of, I'm just sharing part of that. How to tie all that together is really important, and that for us, that takes a pretty much, like I said, a dedicated team who's focused on that, and then that drives our PD team for some continuous improvement. Like over my time at Kite and at PACT, we learned some things about our medias, whether it's in the activation stage or the growing out, that there's differences we saw in medias that really led us to make continuous improvement to get to a stage to be able to scale not only on the dose levels that we're working on, but even if we wanted to go higher, we could go higher. Or if we wanted to shift the percent of, we focus more on the younger phenotype in the final product than having more, less of the cells are just all effectors. Not that effectors are bad, they definitely can attack tumors and kill them. They don't hang around long, they get exhausted. So those are some quick examples.
Thank you, Tim. And when do you consider is the appropriate time to sample your process throughout the development? What do you look for, and when do you think is perhaps too much, right? I mean, in terms of like evaluating the process as it goes on. So what do you think of that?
So for right now, we kind of, we do our, for example, incoming ACE, grab samples. After we've activation, a day after we grab, after we gene edit, we grab, and then midway through our full growth, we grab, and then obviously at the end. That's how we do it, to really see what trends. And again, you can see different incoming patients, how they respond differently, and it may be related to your manufacturing process. It may not be. It may be something to do with the incoming patient. Like I'm sure the panel here has experienced where I had one experience I remember from my Kite days where one of our patients got chemoed like 2 weeks before they got AFed. And to make a long story short, the cells didn't do hardly anything when they came in.
Yeah, it does. Thank you. Thank you, Tim.
Hey, Juan, I think something that you know well too is like some of these cell types don't like to be bothered when they're growing out, right? So
That's
other
right.
ways, other analytics that can be a surrogate, or especially in a commercial setting, in a scalable setting, something you can do without being disruptive to the cells during those certain stages, whether it's again glucose, lactate, other metabolites. And I think, and having the technology integrated into a device where that happens real time. If you think in a commercial setting, taking from the antibody world, where you can now think as cell and gene therapy expands, you're almost thinking about these manufacturing execution systems where you're tracking all of that data automatically. You start to use machine learning and AI to think about how you correlate all of those batches, these tens of thousands of patients. And then like you all talked about as well, the panel has said, looking at the clinical outcome globally and redesigning or making better products later. So some of that technology it's getting there. Most of it's offline, but I know folks are working on that inline, non-disruptive type of analytics as well, which is really an exciting part of the field to think about how we're going to grow globally and expand.
Yeah, that's a very good point. And you talk about not disruptive, which I think at the beginning, people really had a lot of anxiety are the cells growing? And one of the criticisms that I initially heard about the G-Rex is that you couldn't really see the cells under the microscope. You couldn't really control if the cells are growing. And you almost at the beginning needed to take it out of faith that the cells were actually doing well. And I think later on, when you start measuring glucose and lactate, learning really that you can use those as a surrogate. But I think that right, learning when to intervene or mix in the culture, or just let it be, is perhaps one of the things that we also have learned that is actually trying to control the anxiety and just let the culture grow and do its thing is actually probably for the best. Yeah, that's a good point, Mike.
It's better from an overhead perspective, less people, less time, less manipulation, and that's right.
Yes.
And James, what do you have to say on that regard?
Yeah, I was just thinking it would be, Josh is still on the line. It would be great to see a couple pre-sens sensors for pH and potentially DO or something like that on the G-Rex. Something that was maybe wirelessly connected to something. I think that would be a really interesting way to really control that variability and get us a little bit closer to the control we see, in stirred tank bioreactors for monoclonal processes. But going back to a little bit what Tim was talking about, I think, as an allogeneic company, we have the luxury of, choosing our donors carefully, right? So we have criteria that we bring in, only donors that meet, a subset that, is somewhat small subset of the available donors out there to be, to gather a leukopak from. And I will say that even with that criteria that narrows down the population into what we would consider really high quality donors, we still see large levels of donor to donor variability that is clearly donor variability and not process variability. I think it's really kind of been in some ways encouraging to know that our process, when you run the same donor in scale down mode with G-Rex vessels, we get very consistent results among replicates, very consistent results. And it's very clear when our process changes can impact process performance and quality attributes. And I think that, that part's good. That's what, as a process development, group, that's what you want to see. You want to know that you do have some levers in improving process quality. The flip side of that, the more complicated part that we all have to deal with is the variability of the donors, which means that, just because you have levers, you might be starting at a completely different point from one leukapheresis to the next. And, having a platform that's robust across that incoming variation is something that, I think we're all still working on understanding and figuring out how to manage so that the end product is relatively consistent regardless of, that variability.
So Juan, if I may, just to kind of build on James's points, I hear, the terms comparability, I hear the terms platform, I hear the terms analytical development, except that these terms should not be defined. We normally define them for a lot of biologics. What does that mean for comparability when it comes to autologous, right? It's different. What does that mean in terms of platform? Certainly, it's not the platform that we have in mind, let's say, with regard to a map process. Clearly, that's not what we mean. But then what do we mean by that? And then also analytical development. I don't really think that it's analytical development per se. It's really characterization, because the fine differences actually condition your frame of mind in terms of what you're looking for, how you would measure those things. And also just like knowing that we have patient-to-patient variability to start with, assuming that we actually know what those differences mean and how we would control those differences, inevitably would impact how you would demonstrate comparability. Inevitably would impact how you would build a platform to respond to those differences, right? So these are very, very deep scientific questions that we need answers for, that we don't have answers at the moment. I just want to just kind of remind ourselves that we need to change our frame of reference when we begin to try to wrap our arms around how we would exert control over the process as well as the product.
Yes. So Amos, if I can paraphrase, and you can correct me if I said it wrong, you're saying that basically the first thing we need to do is to understand our product, understand how it works, and then from there work backwards in terms of how to characterize the process, how influence that product. But first of all, we need to understand the scientific mechanism of and the principle that how the cells work. Is that what you're describing?
No, not really, because that's a tall order, right? I mean, I think that even just having a good set of relevant characterization measurement of our product during the process, in-process characterization of the various pools, like Tim pulled, let's say four different pools during the proliferation stage. Are we measuring the right thing to see, their relationship to what the starting material variability? Are we measuring the right thing in terms of those 4 pools predicting final product performance, right? The best thing we can do right now is really just correlation. I don't think we're going to claim that we're going to have any understanding of MOA. I certainly will not commit to doing that in a dossier, right? And so really just having the understanding of what are the markers that we're measuring. We know that the typical flow panels don't necessarily give us what we want, but they're a good starting point. I also feel that really what we're trying to understand now really is like, what are the stress markers that we're really trying to measure that would be, let's say, stability indicating, that would be performance indicating. That's kind of what I'm beginning to home in. Like, are there certain stress markers that we can home in on, that we can begin to control? The answer right now is no, but I think we need to get there.
Yeah, I mean, I think that those are very, very good points, right? And I think that I agree. I think at the moment, the best we can do is to try to measure and collect information without necessarily filtering things out, because it's still, I still think we're still learning as a field in terms of what aspects become important. And those things might be different from product to product, right? I mean, I think I would put I guess a voice of to be cautious, right? Because whatever becomes important for a CAR T-cell product is perhaps not necessarily translatable to a TIL product or vice versa, right? And I think that taking those things as gospel can be dangerous, right? So I think that without a doubt, there are phenotypical attributes that are important, but they're not necessarily equivalent from one process to the other. So I think that the best thing we can do at this point is to try to understand as much as possible and collect as much information as possible that will eventually be correlated with the clinical performance. Because outside of that, we're just guessing, right? I mean, what attribute we consider or tag them to be superior, but ultimately are those tags are ultimately going to be correlating with clinical output, and those to be clinical safety or to be clinical outcome in terms of anti-tumor responses, right? So I think that is definitely something that I agree, right? I mean, I think we're still in that sort of like learning phase, right? And Kelly,
I just want to share another experience in terms of non-invasive measurement of product quality attributes and process performance. It's actually kind of ironic that so this is what I meant by the technology itself is going to evolve, right? So I was leading an effort in implementing Raman spectroscopy as a way to measure these various parameters in real time, literally just taking a page out of the monoclonal antibody cell culture playbook. And then what we found out is that while, let's say, G-Rex is great for all the other things that we say that they're good for, right? It turned out that when we try to do non-invasive measurement into the G-Rex cell culture, the signal that we get from that is not as clean as what we would get from a bag, for instance. So there's all these interplay, right, in terms of technology. And as like I said, what I said is that we're trying to aggregate different pieces together. And as we're aggregating them into a system, it has to be in such a way that, when we find something that we need to change out, we need to swap out, that we don't have to go back to the starting point to redo everything. That's kind of what I'm trying to emphasize here is that.
Yeah, no, I completely agree. And I think we all have had that level of experience, right, of how the process changes, and no one wants to go back and redo your clinical study simply because you have to change a critical part of your process. I think as long as you're able to show comparability and you understand your analyticals well enough, hopefully you are able to show that level of comparability and be able to advance the process without reinventing the wheel. Kelly, I would like to hear your thoughts on that before we review the response from the questions from the audience.
Yeah, no problem. I mean, everything that was said was very much in line with things that we encountered over the years. But, there was something that was mentioned, I think it was Mike mentioned, cells don't like to be bothered. That is in many ways sort of a counter. That's a difficult thing to manage when you're trying to grow and scale cells because, without bothering them, how do you figure out whether they're doing okay? So, there's some fundamental things that need to be addressed, and hopefully at some point I'll be able to get my sleeves rolled up and address some of them at some point in the future. But I think that having more systems to monitor cells and closer access to that, I feel like we're getting there, and with systems like. Again, Mike and I are pretty aligned on this in terms of monitoring certain metabolites or growth factors. I think we're gonna get there. But I don't have much more to add than that, other than everything that's been said is pretty spot on with what the industry's seeing. So,
thank you, Kelly. Okay, maybe we can take a small pause and then we can look at the response from the survey. Let me see if I can look at this. So from the question that we asked, what do you consider the biggest challenge when scaling up a manufacturing process? The response that we got is that the biggest thing is the response is 38% for B, robustness of the process, followed by A with 33% for product comparability. So it seems like with the response that we have here is that the biggest challenge becomes in terms of product robustness and product comparability, right? So which is very interesting. I think that's one of the things that in the panel discussion was actually alluded a moment ago. I found interesting the audience considered that the least important was the cost. Maybe it's a reflection that as long as something works, no one really cares about how much to pay for it. What do you guys think about these responses?
I mean, I think that's terrifying that we aren't thinking about cost. I mean, it really is. I think the health insurance systems are going to really, really choke if we don't start paying attention to this. And my 2 cents on this would be, it depends on which department you're asking. So, product comparability would be 98% if you're asking somebody in QC and analytics, and robustness of the process would probably be if you're asking somebody that works in the manufacturing department. But I'll pass to some of the other members for their thoughts.
Kelly, I agree with you. Cost of manufacturing is something that I feel is the biggest challenge, especially when you're dealing with autologous. Allogeneic that, the colleagues on the panel, some of them working on, that's going to help cost of goods. And the cost is really, and I don't have much anxiety over product comparability or robustness of process if you got a good staff who can work on those things.
Yeah, but so can I challenge that? Do you think that response is perhaps a reflection of where you are in the life cycle or of that particular product, right? So if you're early stage and you still have a lot of difficulty with the robustness or reproducibility of your product, perhaps cost is not the most important thing in your radar, right? I think that, for example, Tim, in your case, like you have experience at Kite, and I think those aspects of reproducibility are perhaps behind you, right, in some of those things, right? So I guess once you go to a certain stage, those things might shift, and then cost becomes high in the list. What do you think of that? Do you think it's possible that it's just a reflection of where you are in the development cycle?
Well, I focused in on what's the biggest challenge. And while when you're in early stage, no doubt about it, robustness of the process and product comparability is where you're spending all your time on. No question about that. So if that's being translated to the biggest challenge, then okay, I see where they're coming from. The reason I don't translate to there is, to me, that's normal challenge. You always, in the early stage, got to work through your continuous improvement of your process. It's as you scale up and launch commercially, especially for autologous, how are you going to continuously improve the platform process such that you can get cost of goods lower and lower? As Kelly said, it's still going to be a challenge in the marketplace. And that's why I put it in. So I'm not, let's say, disagreeing with them. I'm just giving you the definition of how I translate the biggest challenge.
Got it. So if I say in a different way, maybe it's okay to be worried about this thing. It's perhaps normal. If you're in early stage, those things will pass, but maybe those priorities will reshift as you advance.
My times at Genentech, we had challenges in robust processes.
But what else, Amos? Do you have something to add?
Yeah, I was just thinking about what Tim said, right? Tim, you say, well, you're not worrying about all the other product quality stuff as long as you have a staff that can handle it. Well, therein lies your cost, right? If you break down the pie chart in terms of what the costs are coming in, at the moment, I think for us, mostly it's, the antivirus is just through the roof, the cost of that, if that's the system we're using. Manpower is a significant part of the cost, besides just raw material. Those are the three major buckets I think is probably known for everybody that are bothered to look at those numbers. So, I mean, it is kind of, it's there, it's staring at us everywhere we look. Every company, every compound has the same sort of cost structure. I would say that some of that are baked in terms of like, as long as we're still using all the same chemistry and biology to do activation and right. Now, the manpower part, Tim, I mean, it seems like you come from that world, and you and I come from that world. We pretty much know how we would, right, streamline that out through automation, through some of the continuous improvement. I just question how much of, when we do what we do, what we know is possible, how much can we bring the cost down? Like what is humanly possible is what I'm still kind of struggling with at the moment. Does anyone have a good sense of like, okay, well, right now it's about X, and then we could probably, by the time everything is well and good, we can expect 50% improvement. I'm not seeing 50% improvement.
Yeah, I mean, from my perspective, I think some very fundamental things need to change in terms of the way the overall infrastructure is set up within the manufacturing organization. I think a good start is having platforms that are started on things like the G-Rex, because then you can work to solve some of the other problems that are out there. If you're still working on what platform you're growing it on, as we talked about before, you're going to be still working on a lot of variability. But I see, having been in the manufacturing world for the last 11 years, and some of you guys in it for way more than that, it seems to me that we're still operating on models that were around in the 80s for this. It's slightly different, but still the core technologies in terms of infrastructure, physical infrastructure, facilities, operational models of manufacturing are from what we did in the 80s. I don't know if some of you who've been around longer than that feel the same way that I do, but we need some really radical changes in terms of infrastructure, manufacturing infrastructure, and the way these manufacturing organizations are run in order to really, achieve cost savings.
Thank you, Kelly.
Just an example. Sorry, Juan. One of the things that excited me when I looked at the Wilson Wolf internet and saw the automation video for the future. That was just, for lack of a better word, a cartoon for right now. It excited me because I sat there and looked at that and said, that can scale out to treat thousands of patients, and I wouldn't have to hire hundreds and hundreds of employees. That's an example of how cost of goods could really come down in this space over time. Just one example.
I think it's through those collaborations too in the industry, like the Scale Ready itself, like the idea of Scale Ready, where it's devices, it's the bioreactor from Wilson Wolf, it's raw materials, and the ease of preparing raw materials and consumables. So these consortiums, whether again Scale Ready, something like a different platform as well, that's going to help. And then that's what's going to empower and enable more closed system processing. And then, as Kelly is mentioning, like the cost of running a plant and reducing overheads and air handlers and grades of classification that we have to manufacture some of these cell therapy, functionally closed or not. You still have to show it and validate it, and I don't think the industry is there yet. So that starts with, I think, these collaborations with different companies that have that technology to be able to do that. And and again, like you said, Juan, before, like where do you do it? Like some companies like us, we've built that infrastructure to do that process development to GMP to help companies do that as well as our own. So it's finding a place that can access that technology, because in some of the small biotechs, it's a lot of cost. It's a lot of cost to build a team like Tim's trying to build and bring in those devices and do that work. So as the industry starts to collaborate at those different technology companies, I think that's a great start. So it's a great idea for the platform itself.
Thank you, Mike. Okay, so before, let's perhaps move to the next two questions to review, if that's okay. Question 2, we just got the survey from the audience as well. The second question is, how would you rate your organization's ability to scale up a process? And here we see that the greatest proportion, for 46.1%, responded C, that they can, but they need to optimize a strategy, right? And it's very interesting that the lowest response was actually for A and D, and which are both extremes. A, for a 15% saying cannot today, need a strategy, and D, which is the other extreme of being ready, can and are confident in our strategy. So it kind of shows a very good spread in terms of the readiness on the audience. Any comment or word of advice after seeing this response from the panelists?
just say it almost follows the survey one sort of, if you think about the audience, it looks like 60 people or so, like the audience, you can almost see that they're going through the same challenges. They have a process and trying to scale it, thinking about comparability, and also how now in that second response to get there. So it's almost the same audience there.
Yeah, that's a good observation. Yeah. And I think from one of the things that I have heard from many of you is a natural process, right? I think that as you start, those are the things that you're going to be in your radar, but those things will pass, and then other things become more important, perhaps as cost and scaling up and out, right? We have about 10 minutes left. I would like to move to the final question. What do you consider the G-Rex bioreactor viable for clinical and commercial scale? For this, we have a 50% response on A saying, yes, we're planning to use G-Rex, and then a 16% spread throughout the rest of the options from D, C, and from B, C, and D. So what do the panelists think about this response from the audience? Yeah. So yeah. I think in general is interesting what we have described, right? Is a general acceptance on the G-Rex. I still think that that it is a system that will be probably suited for many people, but perhaps not everyone. And there are people that are still interested to looking into this technology and incorporating into the process. I think, as Mike mentioned, will be the integration of the Lobo and Biotechnic into the G-Rex platform that probably will make some of these things more applicable in the way that they're integrated into the end-to-end manufacturing process.
I mean, like Kelly said too, cuts the headache out of development for some of these therapies, whether it's autologous or allo using the device. Thinking about having to train someone on something that's a standalone device and needs this or that from a facility perspective, I think it offers a lot of flexibility. It's scalable. So I think it does take a lot of the headache out of development.
Absolutely.
Two Boulder is far easier to train on than managing aseptic pipetting.
Yes, well said. With that, I think, Josh, what if we try to play that video again? I don't know if you have managed to address all the technical issues. Would you like to give it another go?
I was told it did play for folks in the audience, so I think we're good.
Oh, it did.
And I was just going to comment a couple things there on those last couple of questions. I think part of it is, and it was called out earlier, an education process, right? Like the end users in the field that are developing these therapies have so much to be concerned about, huge challenges, as Kelly noted. And there's groups out there like Scale Ready now, other consortiums, or the engineering folks at Wilson Wolf, who are here to educate on the best use of these technologies, and then take that feedback and quickly turn around the advancements in the field. And heard you loud and clear, James. I think the engineers are downstairs working on those inline sensing capabilities right now. But that only works when folks come and partner with groups like Scale Ready and basically say, here's what we need, here's why we need it, and then also listen to us from the standpoint of here's what we've seen work, or go to CMOs that really know what they're doing and that have seen a lot of different processes come through.
Okay, thank you, Josh. And do we have any, in the last 5 minutes, do we have any burning question from the audience? If not, I will pass my last question to the panelists.
I'm not seeing any in the system, Juan, but I don't know if I'm looking in the right place.
Okay. Well, I have a question, and perhaps we finish off with that. And I know we don't have a lot of time. We only have 5 minutes, so I will appreciate it if you could keep it brief in a minute or so. If you have to spread a word of wisdom to the audience in terms of a lesson learned, what would you say in your experience, something to do or not to do? And maybe we'll start with you, Kelly. What would be your advice to people in the audience based on your own experience?
I'm sure this is going to be very similar to everybody else, but just borrow from the experience of those who've done it in the past, and take the development, take the growth platform, which I would say the G-Rex would be a growth platform. Take that out of the picture. You have enough variability in your process and your product, whether it be, as everyone's alluded to, the starting material, the raw materials, QC, analytics. That's not something that really you need to worry about now as the growth platform. Get on that as soon as you can, because you have a lot of other issues to address.
Thank you, Kelly. Mike, what would be your word of advice to the audience?
Don't cut corners on the cost of raw materials. Select high quality raw materials from the beginning, and look at the quality, the grade, and think for the future and plan for success. Develop a process, work with teams that have done it before, as Kelly said, and plan for success of the program that you're working on.
That's great. Thank you, Mike. Team, what would be your advice?
Based on what you're working on, build out a team for continuous improvement, because it never will stop. So you got to have a team that you can leverage their knowledge and expertise to advance the platform as you grow.
Thank you, Tim. Amos, what would you say?
Yeah, follow the product quality attributes every step along the way. Leverage your, translational science group, because they know a lot from the beginning. Don't throw the baby out with the bathwater. They know a lot.
Thank you. James, you're the last. What would be your advice?
Yeah, I think I would echo what everyone else has already said. I think I don't have a ton to add, but I think prioritizing the unit operations that really matter. To kind of echo what Kelly was saying, that you can't do everything, and our process development and analytical teams are not as big as we would like them to be. That's just business. So I think that you got to prioritize. You got to figure out what you can put your efforts towards today that are going to have the biggest impact on the success of your products.
Thank you. I appreciate it. I mean, I think that I agree with all of you. I think that is really important to integrate a platform that it is flexible, that it will allow you to grow and evolve as the clinical programs will do, and to have a top-notch quality team, analyticals, that would allow the process to evolve as the clinical program will do. I really would like to thank you, all of you, for the participation, the engagement throughout the discussion. I personally learned a lot, and it was a pleasure to interact with you and meet you, and looking forward to future, hopefully face-to-face interaction where we can go for a drink and discuss this in more detail. I really enjoyed the conversation. Thank you very much.
Thank you, Juan.
Thank you, Juan.
Thanks, Juan.
Thank you, guys.
Have a good rest of your day.
Take care.
Thank you.
Bye-bye.